Hello aspirants, myself Asmita. Today we are going to discuss about NEET PYQ's 2023 paper biology part. So the question is, which of the following stage of meiosis involved division of centrosome? Option A, metaphase 2, option B, anaphase 2, option C, telophase 2, option D, metaphase 1. And the answer is anaphase 2. Let us see the explanation. The division of centrosome occurs during anaphase 2 stage of meiosis. During this stage, the sister chromatid of each chromosome separates and they are pulled towards the poles of cell by the spindle fibers. The separation of sister chromatid is facilitated by division of centromere, which is the specialized region of centrosome that links the sister chromatid together. Let us see the next question. Upon exposure to ultraviolet radiation, DNA stained with ethidium bromide will show. Option A, bright blue color. Option B, bright yellow color. Option C, bright orange color. Option D, bright red color. So the answer is bright orange color. Let us see the explanation. Upon exposure to UV radiation, DNA stained with ethidium bromide will show bright orange color. Ethidium bromide is a fluorescent dye that bind to the DNA and become brightly colored under UV light. When explosion of UV radiation, the dye intercalated into double helical structure of DNA and imitate a bright orange-red color. This property is often used in a molecular biology and genetic research to visualize and analyze a DNA fragments and agrogel. Let us see the next question. In tissue culture, experiment of leaf mesophyll cells are put in a culture medium to form callus. This phenomenon may be called as dedifferentiation, development, differentiation, redifferentiation. So the answer is dedifferentiation. Let us see the explanation. Tissue culture experiment, leaf mesophyll cells are put in a culture medium to form callus. This phenomenon is called dedifferentiation. It refers to the process by which minor cells lose their particular characteristics and they closely resemble to embryonic cell. In tissue culture experiments, leaf mesophyll cells can be included undergoes dedifferentiation and form callus when they place it in a culture medium that contain right combination of nutrients. Then they will leaf mesophyll cells can be dedifferentiation. That cells in a callus can be included to differentiation and form a new tissue or organ. Let us see the next question. Which micronutrients is required for splitting of water molecules during the photosynthesis? Option A, magnesium. Option B, copper. Option C, zinc. Option D, manganese. So the answer is manganese. Let us see the explanation. The micronutrients required for splitting of water molecules under photosynthesis is manganese. In the light-dependent reaction of photosynthesis, light reaction is used in the photolysis of water molecule into oxygen protons, electrons. In this process, water is converted into oxygen, proton and electrons by the process of photolysis. This process occurs in the thylakoid membrane of chloroplast by the complex of proteins called photosystem II. Manganese is a critical cofactor of photosystem II complex. It plays a key role in binding and stabilizing the water molecules during the photolysis reaction. Let us see the next question. Given below two statements, one labeled as a assertion and another is labeled as a region. Let's see the assertion. ATP is used at two steps in glycolysis cycle. Reason: first ATP is used and converted glucose into glucose 6-phosphate, and second ATP is used in conversion of fructose 6-phosphate into fructose 1,6-phosphate. Choose the correct answer from the following option given below. Option A, both A and R are true. R is the correct explanation of A. Option B, A is true but R is false. Option C, A is false, R is true. Option D, both A and R are true. A is not correct explanation of R is not correct explanation of A. Let us see the answer. Answer is both A and R are true. R is the correct explanation of A. Let us see the explanation. In glycolysis, glucose is converted into pyruvate through the series of enzymatic reaction, and ATP is used in a two steps. First step is used when glucose is phosphorylated into glucose 6-phosphate by the hexokinase using 1 OTP molecules. Second, when the fructose 6-phosphate is phosphorated into fructose 1,6 bisphosphate by the phosphofructokinase using another ATP molecules. So the answer is both A and R are true. R is the correct explanation of A. ATP is used two steps in glycolysis cycle. Thank you for watching. Please like, share and subscribe to Bhagyalakshmi IAS Institute for more videos.